Good afternoon. So yes, my name's Paul Gooley and I'm from the University of Melbourne. And it's my privilege to chair this next session. I must say this has already been an incredible meeting, the diversity of talks. It's hurting my little biochemist's head, to tell you the truth. So the first speaker here is Dr. Leighton Barnden from the National Centre of Neuroimmunology and Emerging Diseases from Griffith University. So I'll turn it over to you, Leighton. And I'm watching my clock.
Oh dear, thank you. Welcome back, and I'm afraid I'm going to wrench you from the microscopic, the performance of cells, to the macroscopic, the performance of organs, in particular the brain, and talk about brainstem myelination and magnetic resonance imaging changes in CFS-ME. So, in summary, I'll just give you an intro to the brainstem, to myelination, and then talk about the methods we used in our MRI studies of CFS-ME. We employed both structural MRI, although we did it differently to most, and functional MRI. And then we'll look at the results and end with conclusions. Brainstem is there at the top of the spine, at the base of the brain. It's got connections basically everywhere. It's made up of the midbrain in red, the pons, and the medulla. So the brainstem, what does it do? It contains multiple small nuclei, they're called, which are just centres or areas, if you like, which send fibres to the cortex, where they release excitatory neurohormones. And they're listed there on the left, and you'll recognise a lot of them. This process maintains arousal and consciousness, and it impacts on cognitive performance and enables initiation of movement. There are also inhibitory fibres that regulate the transition to sleep and the duration and intensity of pain. And it's obviously important to get the balance right. And to do that, there is actually communication between these nuclei. And so it's of interest that this communication is okay, and talk about that as we go. There are other nuclei that regulate heart rate, blood pressure, and so the signals for that come from this vasomotor centre here in the medulla. But those signals are also influenced by communication with the hypothalamus and the cuneiform nucleus in the midbrain. Also, not shown, there are centres that regulate temperature, breathing, gut, and immune function, and more. So these are the autonomic functions. So if transmission of signals within the brainstem is impaired, you can affect problems with arousal, cognitive performance, initiation of movement, quality of sleep, pain and bodily functions. Myelination, this is a wire, if you like, blown up. The yellow bit is the axon, which is a part of a neuron, and it's the long bit which enables the individual neurons to communicate with each other. Surrounding that, there's another cell which contains these circles of myelin. Now, myelin speeds up signal transmission, but more is not necessarily better. It's more important that there's a correct balance between signals coming from different parts of the brain and so on. So we are interested more in not how much myelin there is, but whether there's a difference between chronic fatigue and healthy controls, and if it changes. And we are able to look at myelin because T1, which is one of the basic MRI imaging methods, the contrast of that in white matter, 90% of that comes from myelin. And so when we're looking at our images, we can get an idea of what's going on with myelin.
Information at emerge.org.au is connecting.
Beauty. And myelin is what is white. It's what constitutes most of white matter. So study designs, talking here about structural MRI, and there are several flavours, 3D, T1 and T1 spin echo and T2 spin echo. This is what is mostly used. It looks great, you've got great contrast, but in terms of looking at the signal amplitudes across subjects, it's quite noisy, and we've found that these two, particularly T1 spin echo, which is really not very popular, has given us really quite good results. So here's a brain in pretty colour, which is showing in 3 different planes. It's actually a digital image. There's something like 500,000 voxels in that image. Voxel is like a pixel in a picture. But for the present purpose, I'll characterise with the big cube the volume that contains the whole brain, and the little cube a voxel. So the simplest design is simply to get 2 groups of patients, one being chronic fatigue, the other controls, and compare the groups. And basically that's looking at the values in the test voxel, in the controls and in the chronic fatigues, working out the means, working out the standard deviations, and doing your standard statistical tests. So there's nothing different about what is done at the voxel level, but you do that for all of the 500,000 or so voxels. There's no a priori, you just throw the whole thing at the computer and say, generate this map of the t-statistic values, and I'll look at it, I'll put in a threshold, and so I can see where there's significant difference in the brain. And you know, the important part of this is that you get a location in the brain where that difference might be. We didn't get a lot of results until recently doing just group comparisons, but we did get results doing tests for linear regression. So again, you take your CFS group, look at the values in your test voxel, and you do a linear regression against a clinical score. It might be severity of symptoms, it might be blood pressure, heart rate. And again, the stats will give you a t-statistic for that voxel, whether or not it finds a significant regression. Now, with things like heart rate and blood pressure, you can get values for the controls as well. And so there's an interaction with group, the statisticians call it, study you can do, which for each voxel looks for a regression in one direction in the CFS and in the opposite direction in controls. Now this is really quite powerful, because if you can find voxels where this is happening, you can say that in CFS it's different to normals. That is, it's abnormal. So firstly, let's look at the relationship between our MRI and blood pressure. Is it different, and if so, where? And this is the sort of result we get. So that map, the t-statistic map, what's on the right of the previous figures, is shown here in colour. And in the background, in the grey, is just a reference brain, so we know where we are. And this is a significant interaction with the group result for T1 MRI versus pulse pressure. And it's here in the cuneiform nucleus of the midbrain. These are some transaxial sections. And when you extract out the data for the individuals and plot it, you can see this difference in slope for the two populations. So the triangles are the individual values and the lines are the best fit. In the normal controls, basically what you're looking at there is homeostasis across the population in this particular context. And it's different in CFS. So we then looked again with looking at pulse pressure, but this time T2, we found similar interaction with the group results down in the medulla here, which is in the vasomotor centre, which controls blood pressure and heart rate. So these are really quite insightful results. Now, we also did the group comparisons, and we didn't see anything here or there. So it's probable that these parts of the brain are normal, but what we're seeing here is caused by a problem with communication between the regulatory nuclei in the brainstem. And so in this case, that's what's doing the blood pressure control. If we've got problems here, then we're going to get abnormal signalling, which could culminate in this difference in homeostasis in chronic fatigues. Another correlation we did was with CFS severity, and we found we found significant correlations here in the internal capsule and further on up in the white matter of the frontal lobe. There were two different measures of severity used in the different colours. Now, the interesting thing about this was that the more severe the symptoms in chronic fatigue, then the more myelin there was here, which is counterintuitive. The more myelin, the better the nerve transmission. And so what's going on? And what we think is going on is that this is a compensatory response for weak nerve transmission in the brainstem. The fibres that come through the brainstem go through the thalamus here and through on up to the cortex, the thinking part of the brain, if you like. And it seems that this is a compensatory measure for problems in the brainstem. As a side product of this, we were able to get an idea of the role that anxiety and depression plays in CFS-ME. We were aware that there were some people saying that, it's just a result of depression. This is just the figures that I was showing before. And we actually acquired scores of anxiety and depression using the HADS questionnaire, and we were able to include anxiety and depression in the analysis. We adjusted for depression. The idea being that if this severity was basically proportional to depression, then when you adjusted for depression, it will go away. What did we find? it actually became stronger. The stats became stronger. It's in the same place, but the stats became stronger when we adjusted for depression, when we took that out. And so that means that, sure, there are MRI differences associated with depression, but they're independent of the MRI differences associated with severity. You can see, if we look at a single voxel and plot the CFS severity versus the MRI, after adjustment in blue, you can see that there's a tighter distribution about the line, and that's because the variance introduced by the depression has been taken out, and that's what's give you the stronger stats. You've got a better fit to the line, but the line's the same. Okay, in a more recent study with a better MRI, we actually were able to see differences between groups. We had bigger groups as well. But here in the brainstem is where chronic fatigue myelin levels were lower than controls. But here in the sensory motor area up top, the myelin levels were actually greater than in healthy controls. So what we thought we would do is we'd, for the individual subjects, extract out and plot the values up here versus the values down there for the individual subjects. And when we did that, we found there was a reciprocal relationship between the two. To our knowledge, this hasn't been reported before. And I think the most obvious explanation is that this is a homeostatic mechanism that maintains appropriate signal transmission from the body through the brainstem to the cortex, so that when the brainstem has pretty high levels of myelin, there are lower levels up above. When the brainstem has lower levels, then there are higher levels of myelin near the cortex. The average result being that the signal transmission should be adequate in either case. Now here, it seems that because myelin and signal transmission was impaired in the brainstem, so we're down here, in chronic fatigue, which is the red, then the sensory motor myelin up here was increased. There was a compensatory mechanism. So there's an echo of what we were looking at before there. Yeah, so sensory motor myelin was upregulated to compensate for weak brainstem nerve signals. Now, we've recently finished or got towards the finish of analysing some functional MRI. So in the structural MRI we've been looking at to date, you get a single image for each patient and you look at that across the populations. In the case of functional MRI, you actually acquire a long series of quick scans. We actually acquired 1,100. They're only 0.8 seconds long. There's a cost to doing that in terms of quality, and I'll show you something later. But what you do, you look for areas within the brain, in the simplest case, just a pair of areas. And for each area, you look at the contents from the MRI and produce a time series. And you do that for the other area, and you look to see if the time series or the time course is correlated. So area 1 might look like this, and area 2 look like this. This is an example from another place. But in this case, the correlation is pretty good. So you can see this peak and the troughs are sort of more or less related. And you can get a number for this, mathematically. And when you do see the correlation, it's said that there is connectivity between those two parts of the brain, because the time courses are correlated, and they must be connected, somehow dependent on each other. So what we did was, because of our interest in the brainstem, we nominated some areas and looked at pairs of them to see if indeed there was connectivity, and if in CFS this connectivity was different to controls. Before we did that, we had to do some alignment of individual subjects, and we used a mask. We just used the brainstem region itself to do that, because using the whole brain wasn't giving us good results, partly because the top of the brain and the brainstem are sort of not always at the same angle in different individuals. And so these are the areas we tested for connectivity. So here in the medulla and also in the cuneiform nucleus, areas that we showed before were involved. We did it left and right for both. We also looked here in the subiculum of the hippocampus, because it's been shown there are a lot of connections from the brainstem to this into the hippocampus. So I'll now show you what we're up against. This is just one image of 1100 for one subject, and these are different subjects. You can see some pretty nasty loss of signal here. There's some pretty nasty distortion. In the brainstem itself, it kind of looks different from subject to subject. The redeeming feature is that if you look at more of this subject, then they all kind of look the same. So when you're extracting out the time series, hopefully they're okay and going to give you something useful. And indeed, that's what we found. We found that the medulla region down here, when we computed the connectivity and compared it across the two groups, the connectivity was lower in chronic fatigue between the medulla and the midbrain. Also, it was lower between the medulla and this hippocampal input. It was also different between the left medulla and the cuneiform nucleus. And so these are the p-values. It was quite nicely significant. So just a quick look at other brainstem reports. Reduced perfusion, blood flow via nuclear medicine SPECT has been reported, hasn't been reproduced. Inflammation in the brainstem via PET has been reported, but only in 9 subjects, and nobody else has done it, presumably because of the radiation dose the subjects get, or probably more likely because they can't get controls to put up with the radiation dose. There's quite a few groups have looked at white matter volume reduction at a local level from the MRI, and some have found reduced volume in CFS and some haven't. And we looked at the volume and found differences well, not differences, but a correlation with fatigue duration here in the brainstem. But in another 2 cohorts, we couldn't reproduce that. So this was a problem, it seems, with CFS and MRI studies. But anyway, from what we have done, here are the conclusions. Firstly, autonomic regressions with MRI imply, so this is blood pressure, heart rate, imply that there's communication impaired between different brainstem nuclei. Severity-dependent upregulated myelin in frontal white matter implies weakened nerve signals in the brainstem. And there was a compensatory brainstem sensory motor myelin relationship implying upregulated sensory motor myelin as a response to impaired brainstem conduction. But the word here is implies. So the functional MRI is particularly important because that directly measures connectivity between brainstem nuclei. And we found, in fact, in chronic fatigue, there was reduced connectivity. And this is going to affect multiple regulatory functions and may explain many symptoms of CFS-ME. So our next task is to understand what might be causing damage to the brainstem. Thank you.
Thanks very much. Questions? Mike's coming.
Do the connectivity measures tell you anything about directionality from one area to the other?
No. I think there might be a special way of doing it that can, but not what we've done.
There's been some studies within pain showing overactive parts of the brain when stimulating pain as sort of an overcompensatory measure. Could that be similar to, rather than, I suppose, it being overcompensating for a deficiency rather than sort of a sensitisation?
Could be. But I mean, it's also quite likely that if there is problems with the inhibitory mechanisms from the brainstem, that you'll get, abnormal pain levels and duration.
Okay, so I think for this, to keep us on track, we'll thank Leighton again for his talk.